Welcome to the Royal University of Phnom Penh. This unit is about data analysis and interpretations. In these presentations, I will present you the learning outcomes of this unit, the overall data analysis process and approaches, data interpretations, measure of central tendency and dispersions, and p-value and statistical significance. Upon completion of this unit, you should be able to define data analysis and data interpretations, identifies appropriate data analysis and interpretation techniques for your research project, difference between qualitative and quantitative data and analysis, analyze data gathered from questionnaires, interviews, and observation studies, software packages for data analysis, pitfalls in data analysis, interpretation, and presentation, and you should be able to present your findings. Overall methodologies of data analysis and interpretations, you should state the objective of the surveys, define the target population, define the data to be collected, and defines the variables to be determined, define the required precision and accuracies, define the measurement instruments, and define the sample size and sampling methods, then select the sample. You should understand the differences between population and samples. Population is a large group. Sample just represents the large group. In data analysis process, you should follow the 5 steps. The first step, you should define why you need data analysis. The second step, you should begin collecting data from sources. The third step, you should clean through unnecessary data. The fourth step, you should begin analyzing the data. And the last step, you have to interpret results and apply them. The roles of data analysis. Data analysis is an attempt by the teachers or researchers to summarize the data that has been collected in a dependable, accurate, reliable, and correct manners. It is the presentation of the findings in the manner that has an air of undeniability. Data analysis techniques. In data analysis technique, you should identify themes. You should work inductively and look for the themes that emerge from your data. And you should find out if there are patterns that emerge from the events, responses, or key phrases. Then you have to quote items used in surveys, interviews, or in the questionnaires. Coding is the process of trying to find patterns and meaning in data collected through the use of surveys, interviews, and questionnaires. Coding guidelines. To manage your data in easy way to understand, you should read through all of data and attach working labels to blocks of text. And then you cut and paste blocks of text into small notes so that you can have your data in a manageable forms. Group the notes with a smaller labels. Then revisit the parts of data to see if the label fits. For example, in data 1, you can color them in blue, and data 2, you can color in orange, and data 3, you can color in greens, and data 4, you can color in purples, and data 5, you can color in red. And then you can group them according to the colors. To do this, it would be easy for you to understand or to manage your data in appropriate groups. Analyzing and interviews. Another common forms of qualitative data that are analyzed by action researchers are interview data, most commonly in the form of a transcript from the audio tapes of the interviews. Carefully read the transcript along with your notes from the interview in order to identify the broad themes and categories that emerge from the interviews. Simple qualitative analysis. Qualitative analysis expresses the nature of elements and is represented as the themes, patterns, or stories. Qualitative data is difficult to measure sensibly as numbers. For example, counting number of words to measure dissatisfaction is very difficult. Therefore, in qualitative data analysis, you should notice things, collect things, and think about the things to analyze all the information you have collected. The first step is to gain an overall impression of the data and start looking for patterns. Next comes more detailed work using structures, frameworks, or theories to support the investigations. Patterns may relate to a variety of aspects. For example, behaviors, user groups, phrases, or situations where certain events happened. The roles of data interpretations. Data interpretation is an attempt by the teachers or researchers to find meanings in the data and to answer the question, so what? Data analysis try to interpret the outcomes or findings, while data interpretation focuses on the implications. Data interpretation techniques. Be prepared to demonstrate to others that in spite of the modest scope of your research or studies, that your works have implications that are or may be socially significant. Now let's talk about the introduction to samplings. Sampling is a problem of accurately acquiring the necessary data in order to form a representative view of the problem. This is much more difficult to do than is generally realized. Let's view the video clips below to get more information how to select samples.
Recall that a population refers to the group of things that we want information about, and a sample refers to part of the population that we take out to examine and draw conclusions from. In this video, we will be looking at the different methods of obtaining a sample. But first, let's look at the types of biased samples. Biased samples occur when one or more parts of the population are favored over others. The two types of biased samples include the convenience sample and the voluntary response sample. A convenience sample only includes people who are easy to reach. If this is our population, and a researcher comes along to interview people, then he would only talk to the people that are closer to him to be part of the sample. This is a biased sampling method because not everyone in the population has an equal chance of being part of the sample. Only people that are of convenience to the researcher will be interviewed. Now, a voluntary response sample consists of people that have chosen to include themselves in the sample. So the researcher lets people come to him. This is a biased sampling method because people with a strong interest for the survey topic are the ones who are most likely to respond, whereas the people who don't feel as strongly about the topic may not even care to respond. Remember that a good sample is one that is representative of the entire population, and it gives each thing an equal chance of being chosen. When you have these conditions, you have what is known as an unbiased sample. We will be looking at three different types: stratified random sampling, multi-stage sampling, and simple random sampling. The most basic type of sampling is the simple random sample, also known as an SRS. Since an SRS is unbiased, each individual has an equal chance of being chosen to be surveyed. In other words, to be part of the sample. You can think of an SRS as putting names into a hat and selecting N of them. So if I wanted a sample size of 6, I would select 6 papers and come up to the randomly chosen people to interview them. For a stratified random sample, we take the population and we divide it into something called a strata. Strata refers to the groups of similar people. Within each stratum, we take an SRS and combine the SRSs to get the full sample. For example, we could take an SRS of 2 people from each group so that we get the total of 6 people. A stratified random sample is good for making sure that whoever is administering the sample gets in contact with each kind of group. The last type of sampling is called multi-stage sampling. For multi-stage sampling, we use a combination of 2 or more simple random samples. As the name suggests, multi-stage sampling means you have to go through different stages to find where your sample comes from. For example, if we have 3 groups, stage 1 could be selecting which group will be picked using an SRS. Let's say that I picked out group 1. Then that means I would only look at group 1. Then for stage 2, I would do another SRS to get the 6 random people. We go through different stages of simple random samples to get the actual sample, and this is why this is called multi-stage sampling. I'd like to point out that instead of putting names in a hat, there's another way to pick things randomly. We can use something called the random digits table. The random digits table consists of a long string of random numbers, and it can help us do an SRS. To use it, I would first have to label each member of the population with a number. We have 30 people in this population, so I will label each person from 1 to 30. Notice how I have written 0, 1 instead of just 1. Doing this helps us use the random digits table. Since each label has 2 digits, we will read the string of numbers 2 digits at a time. So let's say I want a sample size of 4. We will use the random digits table to randomly select 4 people. The first number on the table is 19, so person 19 will be part of the sample. The second number is 22, so person 22 will be part of the sample. The third number is 39, but our sample size doesn't go up to 39, so we will ignore it. We will also ignore 50 and 34, but we will keep number 5. We will also ignore 75, 62, and 87, but we will keep number 13. As a result, these are the people we would survey.
Samplings. When you form a sample, you often show it by a plotted distribution known as a histogram. A histogram is the distribution of frequency of occurrence of a certain variables within a specific range. We can display frequency distribution by table of frequencies, histograms, bar graph, or frequency polygons. Interpreting results of experiments. Data interpretation is the process of making sense out of a collection of data that has been processed. These collections may be presented in various forms like bar graphs, line charts, and tabular forms and other similar forms, and hence need an interpretation of some kind. Here, we will learn about data interpretation with the help of many important techniques and examples. We will see how we can make sense out of the graphical data and other forms of it. We shall learn to use it to show the most common questions that present in this section of the quantitative aptitude. Simple interpreting quantitative findings. When we interpret quantitative data, we can use descriptive statistics. Descriptive statistics can be about means, medians, modes, frequencies, range, or standard deviation. Percentages used for standardized data to compare like sets of data. Mean is commonly understood average or just say average. Median is the middle value of the data or middle values. More commonly occurring numbers, or we can say most number in the data. Range is variation between upper and lower limits, or we can say the biggest number and the smallest numbers. Standard deviation is the dispersion of a set of values. Initial analysis is to find averages, outliers, depict any pattern from the graphical representation of data on a spreadsheet. The goal of research is to draw conclusions from the data. Now let's study the table on the right hand roughly. What does the table tell you? Does the table tell you the value of the variables if it is quantitative data, or it is the categories if it is qualitative? And does the table tell you the numbers of observation? Is it frequencies corresponding to the values or to the categories? After viewing the table, now you have to list the frequency of each scores or group of scores in a set of scores below. Study and organize display of a set of data and group the scores how often each different piece of data occurs. Create a similar table for the data here. Frequency distributions. Frequency distributions is a basic technique that provides rich insight into the data and lay the foundation for more advanced analysis. A frequency distribution table. Create a table and list categories of scores along with their corresponding frequencies. List the frequency of each score or group of the scores in a set of scores. Study and organize display of a set of data and group the score how often each different piece of data occurs. Then create a similar table as the previous slide. Here is another example of frequency distributions. Study the table on the right-hand roughly. We can display frequency distributions by table of frequencies. Data in ordered arrays range from 12 to 58. This data could be classified into 4 categories: class, frequencies, relative frequency, and percentage. The data in class can start from tens, but less than 20s. We can start from 20s, but less than 30s, and start from 30, but less than 40s, and so on. Then we can look at the data in order arrays. We want to see how often the score occurs between 10 and 20s, and how the score relative frequency occurs. For example, 0.15, and what percentage in these frequencies. So it could be 50%. Here another examples. We can display frequency distribution by bar graph. Study the table compared to the bar graph. We can also display frequency distributions by frequency polygons. Frequency polygons allow us to display group data. For example, a number of boxes of sweets was opened and the contents was counted. Draw a frequency polygon to illustrate this data. Compare the table on your left hand and the graph on your right hand. Measure of central tendencies. Central tendencies is the first aspect of a measurement to consider. Central tendencies describes the most typical behaviors of a group. To measure central tendencies, we can use the following descriptive statistics such as means, modes, median, or midpoints. The means or average is probably the most commonly used method of describing central tendencies. Study the formula of means, then do the calculation with the statistic provided on this slide. Interpretation of means. When we find mean is relatively close to the highest earned scores, that means student performance is rather similar. Mean can tell us how difficult or easy a test is. When we find the highest earned scores equal the highest possible scores, that means students performed similarly and did well on a test. This means close to the highest scores. When we find mean is close to the highest earned scores, but the highest earned score lower than the highest possible scores, that means student performance appears homogeneous and the test seems difficult. When we find mean is quite below the highest earned scores, and the highest earned score equal the highest possible scores, this means students are heterogeneous and the task is complex. Now let's talk about mode. Dataset example problems. Another indicator of central tendency is the mode. The mode is the most frequently occurring values in the set of the scores. What is the mode of the set of the score present in these slides? The example value of data in a data set examples are given in the table below. Find the means, medians, and modes of the given values in the data sets. Solutions. Value of data sets arranged in a descending orders 1 to 9. Mean of the given data set is its average. Now let's talk about medians. The median is the score found at the exact middle of the set of values. One way to compute the median is to list all scores in a numerical order and then locate the scores in the centers of the samples. Look at the data below. What is the median of set of scores? Midpoints. The midpoint is a set of scores is that points halfway between the highest scores and the lowest score on the test. Study the formulas and do the calculations with the statistic provided on the slides. And what is the midpoints of set of the scores? Measure of central tendencies. Central tendency is related to the means and medians. When we find the value of mean equal medians, it is bell-shaped curve. It means normal distributions. When we find the value of means higher than median, it is a negatively skewed. It means that the distributions tend to the right hands. When we find the means value less than medians, it is positively skewed. It means that the distributions fall to the left hands.
In this video, we will be looking at the mode, median, mean, range, and standard deviation. In the last video, you saw how we can display a dataset using things like histograms, stem plots, and pie charts. We use these diagrams to help us
Measure of dispersions. Dispersion refers to the spread of the values around the central tendencies. To measure the dispersion, we use the following statistics such as range and standard deviations. Study the pictures of the graph and the table of variations. Here is the example of calculation of means, median, and mode. It's the solution of examples. Study the data set values and do your own calculations compared to the values presented on this slide. Measure of variabilities. When we measure variability, we can use range. The range is simply the highest values minus the lowest values. Study the data set of score below. Do your own calculations. What is the range of these data sets? The table of variance may help you to understand this kind of variabilities. Range. The overall range represents the distance between the highest and the lowest scores in the distributions. Thus, if the highest score in the distribution is 89 and the lowest is 11, the range would be 89 minus 11 or 78. Because it involves only the two most extreme scores in the distributions. The range is but a crude indication of variabilities. Its main advantage is that it gives a quick or dotted estimate of variabilities. When you find a range cover about 1/4 of a total raw scales or equals the highest possible earned scores, it means that the student performance is homogeneous. When a range covers about 1/4 or less of the total raw score scales, generally reflects homogeneous performance. If a test contains 40 items and the range is about 10, then the group can be considered homogeneous. A range that covers about 1/3 of raw score scales generally indicates heterogeneous performance. A range that covers about 1/3 of raw score scale generally signals a very heterogeneous group performance. For a 40-item test, a range of about 13 or 1/3 of 40s would reflect somewhat heterogeneous group performance. A range of 20s would reflect very heterogeneous performance. Standard deviations. The standard deviation is another way of showing the spread of scores. It measures the degree to which the group of scores deviates from the means. It shows how all the scores are spread out. It gives a fuller description of set of scores than the range, which simply describes the gaps between the highest and the lowest marks and ignores the information provided by all the remaining scores. Standard deviation is the square root of the average distance of the score from the means. The higher standard deviation, the more dispersed and more distributed are the test scores. Let's study the formula of standard deviations. You may need it for your own calculations. Simple example of calculating standard deviations. Let's say we wanted to calculate the standard deviation for the amounts of earned scores in a department of biology have. There are 100 students in a biology department. In statistical terms, this means we have a population of 100. If we know the amount of score earned of 100 students earns, we use the standard deviation equations for an entire population. So what if we don't know the amounts of earned score each of the 100 students have? For example, we only had enough time to ask 5 students what score they have. In statistical terms, this means we have a sample size of 5. And in this case, we use the standard deviation equations for a sample of a population. Look at the examples. Do your own calculation to find out if you have the same results of your own calculations. Interpreting the standard deviation. We should remember that large range means large standard deviation. Large range and large standard deviation, it means that the performance is heterogeneous. Narrow range and narrow standard deviations, it means that the performance is homogeneous. To determine the variability of group performance, we need to focus on, first, a standard deviation close to 1/2 of the range indicates that the group performance is very diverse within the range. Second, when a standard deviation close to 1/3 of the range reflects the scores that are dispersed throughout the range. Third, when a standard deviation spanning 1/4 or less of the range indicates that most scores are clustered within an area. Here you should know that different score patterns mean different standard deviation values. For example, if the range were 20s, an observed standard deviation approaching 1/2 these values, or 10s, reflects two diverse groups in the class. An observed standard deviation of around 6.6, or 1/3 of the range, reflects a more even distribution of scores throughout the range. However, a standard deviation of about 5 or less, 1/4 of the range, reflects a majority of scores clustered within an area. The standard deviation of normal distributions. The total areas under the normal curve represents all of the scores in a normal distribution. Inside the curve, the means, medians, and modes are identical, so the means fall at the exact center of the curve. It thus is also the most frequent scores in the distributions. Because the curve is symmetrical, 50% of the scores must fall on each side of the means. Here are some important facts about the normal distributions. 50% of all the observation, score, etc. fall on each side of the means. Look at the graph or picture here to find the interpretation of the normal distribution. Normal distributions. In any normal distribution, 68% of the observation scores, etc., fall within one standard deviation of the mean. Half of this, it means 34%, fall within one standard deviation above the means, and the other fall within one standard deviation below the mean. Another 27% of the observations fall between 1 and 2 standard deviation away from the means. Hence, 95% or 68% plus 27% fall within 2 standard deviation of the means. In all, 99.7% of the observation fall within 3 standard deviation of the mean. Interpreting standard deviation. 50% of the scores in the distributions must lie below the mean. 34% must lie between the means and plus 1 standard deviations. Therefore, 84% or 50% plus 34% of the scores in the distributions must be below 1 standard deviation. Study these pictures. It could give you some ideas about the standard deviation in a normal curve. Let's study the picture carefully. See the difference in standard deviations. The standard deviation for the boys and the men in the basketball teams. The larger standard deviations and the widespread of the boys, and the closer or the smaller standard deviation is the similar heights of the men. General shapes of test score distributions. A test score distribution reflects one of the common observed shapes. It can be systematical, skews, unimodals, bimodals, or multimodals. Systematicals. The normal distributions or the bell-shaped curve means most scores are near the centers. The lows and the high ends of scales contain a very small frequency of scores. The U-shaped distributions means a large number of scores occurs at the lows and the high ends, but none of U's occur in the middle of the distributions. The rectangular distributions means an equal number of students receive each score. Skew. Negative skews. A mysterious curve means most score occurs at the highest end of the scales, indicating that more students receive higher scores than lower scores. Positive skews means most scores appear toward the lower ends of the score scales, indicating that the test was difficult for most of the class. Unimodals means only one score appears most frequently. Bimodals mean that two scores, some distance from each other on the scales, have higher frequencies than the score surrounding them. Multimodals means more than two scores have optically high frequencies compared to the scores around them. The differences between p-value and statistical significance. What exactly is a p-value? The p-values or probability values tells you how likely it is that your data could have occurred under a null hypothesis. It does this by calculating the likelihood of your test statistics, which is the number calculated by a statistical test using your data. P-values are most often used by researchers to say whether a certain pattern they have measured is statistically significant. Statistical significance is another way of saying that the p-value of a statistic test is smaller enough to reject the null hypothesis of the test. How small is small enough? The most common threshold is p-value less than 0.05. That is, when you would expect to find a test statistic as extreme as the one calculated by your test only 5% of the times. But the threshold depends on your field of studies. Some fields prefer thresholds of 0.01 or even 0.001. Examples: reporting the results. In our comparison of cat diet A and cat diet B, we found that the lifespan on diet A means equals 2.1 years. Standard deviation equals 0.12, were significantly shorter than the lifespan on diet B, means equal 2.6 years, standard deviation equals 0.1, with an average difference of 6 months. THT equals a minus 12.75, where the p-value lower than 0.01. Let's view video clip below to get more information about p-values.
Hey everyone, so today I will be talking about p-value and what does it represent in statistics. Statistics is everywhere. If you see this touchpad carefully, what you see is a normal bell-shaped curve. Let's say p-value in this region is 0.01. This means I touch over here 1 in 100 times. Let's say the p-value in this region is 0.8. This means 80 out of 100 times I'm gonna touch in this region. Now if we see the statistical definition of p-value, then p-value is the probability value for the null hypothesis to be true. To understand p-value, first we should try to understand null hypothesis. The null hypothesis is the hypothesis which treats everything same, everything equal. Let's take an example where plants are treated with fertilizer A and fertilizer B. The null hypothesis here will be there's no difference in the effect of fertilizer A and fertilizer B. Now there are many statistical tests, say for example t-test, f-test, ANOVA, etc. And the final result of each test is judged on the basis of the p-value obtained. The p-value can take any values from 0 to 1. Let's say p-value is 0.1. This means if I repeat an experiment 100 times, 10 out of 100 times the null hypothesis will be true. Let's say p-value is 0.3. This means if I repeat the experiment 100 times, 30 out of 100 times the null hypothesis is gonna be true. Let's say p-value is 0.05. This means if I repeat the experiment 100 times, only 5 times the null hypothesis is gonna be true. Okay, let's take an example of t-test. The t-test is used for the comparison of mean. Let's consider the previous experiment where the plants were given fertilizer A and fertilizer B. Let's say a graph represents the mean and the standard deviation in the height of the plant. Now the t-test is going to compare the two means. Let's say the p-value for this test is 0.03. This means if I repeat this experiment 100 times, only 3 out of 100, the null hypothesis is gonna be true. Now the next question that arises over here is, should we consider this significant or not? Taking a decision based on p-value is completely in the hands of the experimenter or the scientist who is doing the experiment. The researcher decides the level of significance for his experiment. Let's say the level of significance decided by the researcher was 0.05. Then in this case, the researcher have to reject the null hypothesis. And conclude that there is a significant difference between fertilizer A and fertilizer B. Now let's say the level of significance decided by the researcher was 0.01. Then in this case, the researcher have to accept the null hypothesis and conclude that there is no significant difference between fertilizer A and fertilizer B. So to summarize, the researcher first decides the level of significance, then he does the experiment and finds the p-value. If the p-value is more than level of significance, then the null hypothesis is accepted. And if the p-value is less than level of significance, then the null hypothesis is rejected.
To understand this lesson, you have to do this work. The following outline summarizes the procedure used to describe and evaluate a group test performance. A. Describe and compare test results. 1. Create a frequency table or frequency polygon of the test scores. Compares the location of the highest and lowest earned score with the highest possible scores and decide whether they are reasonable given the complexity of the task measured by the test and achievement characteristic of the group. 3. Calculate and compare measure of central tendencies. A. Calculate the mean and noted position in a score distributions. B. Locate the medians and note its position in the score distributions. C. Locate any modes or modes and note its position in the score distribution. D. Compare the means, medians, and modes and describe the distributions as being systematical or skewed based on these measures. Fourth, compare the observation measure of central tendencies and their location in the score distribution to those you would expect. Give the complexity of the task and the achievement characteristic of the group. 5. Calculate and compare the measure of variabilities. A. Calculate the range. B. Calculate standard deviation. 6. Compare the variabilities measured within those you would expect given the complexity of the task and the group characteristic. Note any discrepancy between measures you expect and those you observe. B. Try to explain any discrepancy you find. Discrepancies are usually caused by misjudging the quality of the interaction or the test. They are also potentially related to the misinterpreting the complexity of the task or the group achievement characteristic. Check the group performance as being better than expected as SEPSA lower than expected. Thank you for your attention.